Hello, this is Gina again with another histology lecture. This is your third tissue type, nervous tissue. This is a beautiful picture on the first slide showing a brown neuron that has many processes emanating from its cell body, which is right here, and a nucleus with a nucleolus. Now there are also some other cells localized around it that aren't quite as prominent. Some of these are the same type of cell, others are a different type, and we're going to talk about the different types of cells in nervous tissue. First, let's just discuss where we find nervous tissue, which will probably be obvious to you, at least in some cases. The brain, nervous tissue's there, the spinal cord, and nerves. Now we have two kinds of nerves. They function similarly. They're carrying impulses to or from the central nervous system out to the rest of your body, or back from your body. There are cranial nerves that are attached very close to what we think of as the brain. It's actually the brain stem. And we have spinal nerves that are attached to the spinal cord. But nonetheless, they're both nerves. And then also in the periphery, we have these bulges, which are very difficult to see, and you'll get to see them with the nervous system in more detail. But these bulges next to the spinal cord, as well as different locations throughout the body, these bulges of nervous tissue are ganglia. And the ganglia are just aggregates of neuron cell bodies that aren't in the brain or the spinal cord. So first, our task is to talk about nervous tissue and what's in it. So nervous tissue has various types of cells, and anytime you see this circle with the nucleolus in it, that's the cell nucleus. Okay, so in neurons it's most obvious because you can see some cytoplasm around the nucleus. Some of these other cell types, it's not clear whether you're looking at a whole cell or a nucleus. In this case, you're looking at a nucleus. But nonetheless, you'll probably identify nervous tissue just by the presence of these neurons. Okay, very elongated cell processes on neurons. Now let's talk about a nerve for just a second. A nerve is actually an organ. Nervous tissue is present in it, but it has other types of tissues as well. So this picture shows the cross-section through a nerve. So within it, we would see nervous tissue if we looked under the microscope. What I can tell you is that a neuron has been cut when this nerve was transected, and not just one neuron, but heaps of neurons. So each individual little tiny circle is one of the processes in a neuron. Okay, one of those long extensions. We call those axons typically. Generically, it's a process because it sticks out. So all of those are neurons, and many neurons are found within a nerve. Now, surrounding all of these axons is a connective tissue, and it's like a sheath. It's a protective layer, and it keeps the axons bundled. In the central part of the nerve, you see a blood vessel with a wall of the blood vessel. So inside the blood vessel, colored red in this picture, you see blood. That's a type of connective tissue as well. But in the wall of the blood vessel, there's smooth muscle tissue so that it can dilate or constrict, become narrower or wider, and epithelial tissue will line the interior, the lumen of the blood vessel. So the bottom line on this is that we consider a nerve an organ because it has multiple tissue types: nervous tissue, couple of connective tissues, epithelial tissue, and muscle tissue. So the cells that are found in nervous tissue are of two types. which I've alluded to already. Neurons are the ones that you'll notice right away. This is a cross-section through the cerebellum of your brain, and it shows you cells of nuclei of neurons with the axons extending outward. So those are neurons, but as you can see, there's a lot of other black dots around here, okay, without seeing processes emanating from them. Many of those are a different kind of cell that we collectively would call a neuroglial cell or just neuroglia. Sometimes people call them glial cells. I call them glial cells or glia. These are just non-neurons. Any cell that's in the nervous tissue that's not a neuron is a glial cell. And there are 4 different types: Schwann cells, oligodendrocytes, astrocytes, and microglia. Let's first look at the structure of neurons. So, a neuron, schematically anyway, is made up of a cell body that contains the cytoplasm and the nucleus. And then it has extensions that we call processes, or a process. Some of them are short, some of them are long. Some are called dendrites, those are the short ones, and the axon is the long one. Now at the end of the axon there are branches, and at the very end of the branches is a location called an axon terminal, and that's where a neurotransmitter would be released. Now, it's not always the case that dendrites are short and axons are long. The way that we tell the difference between a dendrite and an axon has to do with which way an electrical signal is traveling. So a dendrite is always carrying the electrical signal towards the cell body. And an axon is always carrying an electrical signal away from the cell body. And we'll see various types of neurons when we study the nervous system. So the functions of these different cell types, I've alluded to with the neurons already, include the use of chemical and electrical signals to communicate with other cells. That's what neurons do. They use electrical signals, but also neurotransmitters, which are chemicals, to help control a target cell. Schwann cells and oligodendrocytes have a similar function. They both insulate axons to make sure that electrical signal does not dissipate. It stays within the axon. The only difference between these two is well, not the only difference, but the major difference in these two is where they're located. Schwann cells are in peripheral nerves, and oligodendrocytes are located in the brain. Now, astrocytes have a function that just supports neuron health, repair, or growth. So astrocytes help nervous tissue because if neurons are transected for some reason, they may be able to regrow with the help of astrocytes. Schwann cells also help with that. They also help control, astrocytes also help control the chemical environment in which neurons exist by membrane transport mechanisms, making sure that the extracellular fluid has the correct concentration of certain ions and nutrients. Microglia provide immune protection. So they're the phagocytic cells that can chew up debris of any damage or maybe fight an infection through phagocytosis. So I just wanted to show you a little difference between oligodendrocytes and Schwann cells, as well as a little bit about the difference in structure of all these glial cells. You're not responsible for identifying these different cell types, but I thought I would show you kind of a schematic diagram so that you can see that they are actually different in structure. So the yellow cells are neurons in this picture. The blue cell is an oligodendrocyte. And notice that it is wrapping part of its cytoplasm, but mainly cell membrane, around the axon of a neuron. In fact, it's doing double duty here. It's actually helping the axon of two neurons. So that's an oligodendrocyte. They form this protective covering on the axon, which is called the myelin sheath. Now the other glial cells are astrocytes, which have many processes emanating off of the cell body. Usually you don't see one longer than the other, any other. And then there are microglia, which are smaller, and they're shown as brown in this picture. Now, what you can't see here is a Schwann cell, and that's because this diagram is actually from the brain. So in the brain, you only find oligodendrocytes. You won't find Schwann cells. Let's look at what Schwann cells look like. They're found in peripheral nerves, and they do a very similar function. So they wrap around the axon of a neuron to insulate it electrically. Now, a Schwann cell gives its entire body over to one axon. So this is 3 different Schwann cells coating the axon or covering the axon of a neuron. Notice the oligodendrocyte doesn't devote its nucleus to the role of wrapping around an axon, and that enables it to send out extensions that will serve multiple axons. So the differences between Schwann cells and oligodendrocytes are where are they located and how many neurons can they myelinate. A Schwann cell can only myelinate one neuron. An oligodendrocyte, many neurons. More than 2 usually, even though these diagrams show 2. That's all for nervous tissue. Thank you very much.